12, we're going to take the stuff we learned about the nervous system and nervous tissue in general and look at the individual parts of the nervous system, starting with the central nervous system. The central nervous system is the brain and spinal cord. And the brain and spinal cord are common to a lot of animals in the evolutionary scheme of things. And the whole process of developing a brain and spinal cord is referred to as cephalization, or the formation of a head, literally translated. And this has happened in increasing, or led to increasing number of neurons in organisms until it reached its highest level in the human brain. At least that's what humans are touting. So if we can understand the steps of cephalization, the evolutionary and developmental things about the brain, it might give us a better understanding of the individual parts when we talk about those. So we're going to start off with how the brain develops. It starts off as in the embryo as a neural tube, literally a tube of cells and tissues that will eventually become our nervous system. The anterior end of that is going to expand, get bigger, and constrict and form three vesicles. Those vesicles or regions are called the proencephalon, or forebrain, the mesencephalon, or midbrain, the rhombencephalon, or hindbrain. And behind that, the posterior end is going to become the spinal cord. So this is very early in development, but these things are going to develop into the adult brain if we just follow them through, including the ventricles. So the primary vesicles, those three vesicles, are going to give rise to 5 secondary brain vesicles. The forebrain will become the telencephalon and the diencephalon. The midbrain is not going to divide, that's going to stay in place, but the hindbrain is going to divide and become the metencephalon and the myelencephalon. Telencephalon will give us the 2 cerebral hemispheres, which will be the first section of the brain we talk about. And together, those cerebral hemispheres are called the cerebrum. I will apologize right now because I will sometimes slip up and say cerebellum when I mean cerebrum, and cerebrum when I mean cerebellum, but try to keep them straight. The 2 cerebral hemispheres, the outermost part of our brain, what you can see if you take the skull off somebody, is the cerebrum. That diencephalon is going to become some things that we have talked about and will continue to talk about throughout the year: the epithalamus, the thalamus, the hypothalamus, and the retina, part of the eye. Your eye really is an extension of your brain. The mesencephalon will become the midbrain, or remain the midbrain. The metencephalon is going to be the pons and the cerebellum, and I do mean cerebellum. The myencephalon will become the medulla oblongata, and the central cavity of the neural tube within this region is going to become the ventricles of the brain, the spaces of the brain that are filled with cerebral spinal fluid that we'll talk about. So we start out with a simple tube, a neural tube. These are neural crest cells. This is going to become nerve tissues. That will expand in the front end, giving you the prosencephalon, or forebrain. There'll be a little constriction, there'll be some expansion in the middle a bit, and that's the mesencephalon, or midbrain. And then behind that, it will expand a little more, showing these constrictions on either side, to become the rhombencephalon. This will continue to become the spinal cord. This structure, in the next visible phase of development, will sprout out 2 hemispheres, secondary brain vesicles called the telencephalon. This will also expand in the back, come out the side a little bit to give the diencephalon. The mesencephalon expands but doesn't bulge anymore, doesn't really change much morphologically shape, at least not from my perspective. And in the back, the hindbrain, the rhombencephalon, will get 2 bulges, what will be the metencephalon and the myelencephalon. And while this might sound just rudimentary, or maybe why do I need to know this, you probably don't need to know this other than for homework questions. But all of these structures become important structures that we're going to have in the adult brain that we are going to be talking about in the central nervous system chapter. So that telencephalon is going to become the cerebrum, the cerebral hemispheres, which will contain a cortex, white matter, and basal nuclei. The part below that is going to develop into the diencephalon, and we'll talk about the thalamus, the hypothalamus, the epithalamus, and these grow out to be your eyes, your retina. Down here, the brainstem. Brainstem is going to be very important. Part of that brainstem is also from the metencephalon, the pons part. The cerebellum is going to be in the back. We'll look at it when we look at the adult brain. Very important for movement processing, and we think critical thinking even more important than we used to think. And down here we have the brainstem, which will be the medulla oblongata, which will merge into the spinal cord at the point where the brain leaves the foramen magnum in the skull. There are also the canal regions of this neural tube will give us the lateral ventricles, the third ventricle, structure called the cerebral aqueduct, which connects to the fourth ventricle, and then the spinal cord still has a central canal running through it. So it's a way to look at where these adult structures came from, giving you a little bit of information in the background. So the brain grows faster than the surrounding skull. It takes up more space than there is in the skull. In order to do that, it has to fold. So even though we make it look nice and linear here, all of that's happening under a process of folding. The forebrain is going to move towards the brain stem, become the midbrain, pons, and medulla oblongata. Cerebral hemispheres are going to double back on themselves, envelope around the diencephalon in the midbrain, creasing to increase surface area to get all of that cerebral hemisphere where we do our higher level thinking and processing, more space in a confined area. So we have take that linear diagram we had before and try to imagine this happening as a folding. So not only is it expanding this way, it is also expanding this way. And by week 5, we have 2 major flexures, the telencephalon, diencephalon angled towards the brainstem. This is going to continue to grow up and over, as you see here, giving us our cerebral hemispheres. And now by week 13, we start to see a structure that looks similar to the adult brain. The adult brain has 4 major regions: the cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. Some important structures within the brainstem that we'll look at individually as we go through this are the midbrain, the pons, and the medulla. So here is our adult brain. This is what we think about when we probably think about a brain. You took the top of a skull off. This is what you'd be looking at. Those are cerebral hemispheres. Very important for critical thinking, intellectual processing, different regions for different things. Then buried deep within that is the diencephalon. So this takes up the most space. The diencephalon is here. Cerebellum is back here. We'll look at that more closely. And then the brain stem, including the midbrain, pons, and medulla oblongata. And this point here where the green goes to the spinal cord is the point where the medulla oblongata leaves the foramen magnum in the skull and heads down the spinal column. So brain is divided into a number of different ways and organizational things to think about to give you an idea of what's going on. Gray matter are basically cell bodies, the neuron cell bodies. They're not myelinated, so they look kind of grayish when you first look at them. Those cell bodies are where processing is going on. White matter is myelinated, but there are also non-myelinated axons within the white matter. Myelin has a whitish color to it. The basic pattern you find in the central nervous system is a central cavity surrounded by gray matter with white matter externally to the gray matter. That's going to be a little different in the brain itself. And the spinal cord has that basic pattern, but the pattern changes as you go into the brain stem. Into the brain stem, we're going to find more gray matter on the outside than on the inside, whereas on the spinal cord, the gray matter is deeper inside than outside. So if we took a cross-section of the spinal cord, we would see gray matter. These are cell bodies. They are communicating through myelinated axons out to the rest of the body from the spinal cord. As we go into the brain, most of that gray matter will be found on the surface of the brain. But there are places within the brain stem and within the cerebrum itself that have gray matter areas, and we're going to call those collections of cell bodies nuclei. The cerebral hemispheres and the cerebellum, their outer layer is gray. That's called the cortex. That's where all the processing that each of them does happens, or is at least coordinated. And they are communicating with the other parts of the brain using tracts, which are white matter. So the cerebrum, cerebellum also have embedded within their white matter some nuclei that we'll talk about. So if we took the brain stem, which still kind of gray matter around the central cavity, white matter extending out because it is communicating with other parts of the brain and the body, but there are isolated places where you find gray matter patches or islands, and those are called nuclei. We go up to the brain itself, or what we took a cross-section, we would see that in the cerebrum, the outer region is gray. That's where the processing is happening, communicating to the inner regions, and there are places within the inner region that have gray matter islands, again, that we'll call nuclei. And you notice these folds inward and extensions outward. That's the folding that provides us with an increased surface area to get more gray matter. Within that, there are fluid-filled chambers. They are continuous with one another, so there's not, we're going to call them different names, but they are all connected with one another by canals. They're filled with cerebral spinal fluid. They're lined by epidermal cells. We talked about epidermal cells in the last chapter. They are one of the neuroglia cells. There are a pair of lateral ventricles that are the largest. They have kind of a C-shaped look to them. They're deep in each hemisphere. They are separated from one another by the septum pellucidum, which is a membrane. Each lateral ventricle is connected to the third ventricle through the intraventricular foramen. That third ventricle is within the diencephalon. The third ventricle is connected to the fourth ventricle through the cerebral aqueduct, and the fourth ventricle is found within the hindbrain. They are all continuous with the central canal of the spinal cord. There are 3 openings connecting the fourth ventricle to the subarachnoid space that we'll talk about when we talk about protective structures in the brain. They are the paired lateral apertures in the side walls and a median aperture in the roof. So these blue show us from the side view the ventricles. Here's the lateral ventricle, the third ventricle, the fourth ventricle. You can't see this. The cerebral aqueduct goes between the fourth ventricle and third ventricle. And then this is from a different view. And that pretty much covers the development of the brain and why it's important and why it might be relevant for you to get a grasp on, and some of the ventricle structures. And now we're going to look at the individual parts of the brain in more detail, starting with the cerebral hemispheres. But I'm going to stop there for now.